Hello, everyone. I think we will go ahead and get started with the second session of the Koch Institute Summer Symposium. And I just want to provide a couple of announcements for the session. Please silence your phones. You don't want to hear noises if you're taking pictures. You can take pictures as long as it's not on the slide, as long as it doesn't say post. And the other announcement is that for questions, please use the 2 microphones that we have in each side of the auditorium. Thank you. And it is my great pleasure to co-chair this session with my colleague Anders Hansen. And we are in for a very nice treat with 5 spectacular speakers. So we'll have 2 speakers first, then a coffee break right outside. And then we'll have the last 3 speakers for today's symposium. And it is my great honor to introduce Dr. Paul Mischel. And he's coming from the other side of the country, the West Coast, where he is a professor and vice chair for research for the Department of Pathology at Stanford Medicine. And he's also affiliated and an institute scholar in Saraf and ChemH. He is also a faculty advisor for experimental biology at the Innovative Medicines Accelerator at Stanford. And Dr. Mischel is a physician scientist trained in pathology and cancer biology. And his research has not only opened up new ways of thinking and also treating cancer, it's also revolutionized the way that we think about cancer genomics. And we are in for a very nice presentation on novel mechanisms of how chromosomal DNA can be propagated across cells and how cancer cells can exploit these mechanisms. Please help me welcome Dr. Mischel as our next speaker for the symposium.
Thank you. Thank you, Matt. Thank you, Mike. It's really a pleasure, really an honor to be here in this historic location. Just by way of my disclosures. We depend on maps. When we have more information than we can handle, we make maps to help us navigate the complex landscape. Without our maps, we're lost. But if something's wrong with our maps, we're really lost. Have a look at these 2 maps. You see on your left side here, that was Ptolemy's map of the solar system. In the days of the ancients, astronomers looked up at the night sky and they made very precise measurements of the planets tracking across the heavens. And they made a map, and the map was dead wrong, even though the measurements were absolutely correct. It took 1,400 years until Copernicus came along and straightened out the map based upon exactly the same precise measurements. I'm telling you this because we're all modern-day cancer cartographers. We make maps. We can now take DNA from a tumor, and we can map the genomes of cancers because we have a map of the human genome. And I'm going to tell you about something that we found in which the measurements were right, but the map was wrong, and it's very consequential. And it started with a problem of intratumoral genetic heterogeneity. At the time, we were studying glioblastoma, this highly lethal brain cancer. It was known to be very heterogeneous. It was work from David Nathanson, at the time a graduate student in my lab, who's now a professor at UCLA. And we wanted to understand something. People knew that heterogeneity is a problem. If you look at this immunohistochemical slide, this is showing the EGF receptor mutant protein that's highly amplified. The gene is highly amplified in this tumor quite frequently. And notice that some cells have a lot of it, some cells have very little of it. In the schematic, it goes from yellow to red. And Peter Knoll put forth this idea of clonal evolution, that you would select for the cells that are most resistant to a drug over time. And so a wonderful idea, but difficult to test until you had single-cell technologies. So once they became available, we started testing that. And we were in for a couple of surprises. The tumors changed the way that you would expect them to change. That is, you got rid of the cells that were more sensitive, and you got the cells that were more resistant, and you removed the drug, and it went back, except the kinetics were all wrong. It was much too fast. The second thing is that everything we've learned about classical genetics is that mother cells would give rise to daughter cells that are genetically like them. So if you took a cell, a yellow cell in this example, it had very little of the amplified receptor, or a red that had a lot of it, you put the yellow on a dish or in a mouse and you'd get a yellow tumor, the red and the red. But that's not what happened. Every cell could give rise to the tumor with its original spectrum of heterogeneity, and that made no sense by classical genetics. So we ended up doing a critical step here, which was we looked, because I realized that we actually don't know where the DNA is when we actually look at cells in interphase, because the DNA is a complex three-dimensional jumble that determines how genes are expressed. You can look when cells are getting ready to divide, and you can look at the chromosomes, and that's what we're looking at here. The blue are the chromosomes. This is from a patient's glioblastoma tumor. The blue are the chromosomes stained with DAPI, and the red is the oncogene. First of all, notice how many copies there are of this EGF receptor oncogene, and then notice that none of it is on the chromosomes. And then you treat with an EGF receptor inhibitor, and the DNA disappears, and then you remove the drug, and it reappears all in this extrachromosomal DNA. That's a very strange statement I just made. You're treating with a tyrosine kinase inhibitor, and the DNA is disappearing quickly. So I began to really think about this, and I realized that this was the tip of the iceberg. We published this in Science in 2014, and I'd go give talks, and people would say, the oncogene I study, Nick, it's on chromosome 8. And I'd say, how do you know? And I'd pull out a cancer genome map, and all I could think of was Ptolemy's map of the solar system. Do you actually know where genes are amplified? Do we really know? And I went back to the original literature, and it was not at all clear how this was actually happening. Well, how do you get 60 copies in a tumor? None of this made sense to me. So, in fact, people had seen extrachromosomal DNA called double minutes, although in fact most of them aren't double, all the way back to the 1960s. And beautiful work by Robert Shimke and others had shown that this was really interesting, but it was thought to be very rare, 1.4% of all cancers. And why was it that cancers with oncogenes amplified didn't respond to the same drugs when those drugs actually were targeting the mutations of those genes when they were on chromosomes? So I began to think this was a much bigger problem and realized we need to actually interrogate where amplified oncogenes are. I began working with Vineet Bafna, a really outstanding computer scientist at UCSD, and this was work from Kristen Turner, at the time a postdoc in the lab. And the idea was, could we actually integrate the old with the new? Vineet developed a tool called Amplicon Architect that could allow us to look at whole genome sequencing and ask, did an amplicon arise as a circle? And then we would also look by making metaphases in a huge number of cancers with a whole variety of different histological types. And the surprise was that all of the oncogenes that we think of that are amplified are frequently on these extrachromosomal DNA particles. So we published this in 2017. And then I think people began to take notice. And then people thought, how is it that we actually missed this? So I began to think about this. The real issue was that in the old days, people actually used to look at chromosomes to interrogate genomes. So you had spectacular spatial resolution, but almost no genome resolution. And then the way we looked at cancer changed because first there were arrays and then next generation sequencing. So we're actually taking short strands, short reads of DNA, and we're mapping them back to the reference genome. But the reference genome comes from what? It comes from normal cells. So we're actually inferring the location and realized that probably wasn't right. So we needed to look at this. And this is really why it actually matters, because maybe you'd say all that matters is how much of the gene there is for how much of the protein there is. But this diagram illustrates why that's not true. Let's just say you had 3 copies of genes on chromosomes and cells getting ready to divide, it would make 6, and each daughter cell would get 3. But if it's on these extrachromosomal DNA particles and they don't have centromeres, those 3 could become 6, but they could be parcels 6 and 0 or 5 and 1 or 4 and 2 or 3 and 3. Notice 2 things. 1, you've doubled the potential copy number in one cycle. Keep playing it forward. That's how you get 60, 70 copies. And 2, you've increased variance, which Charles Darwin taught us was the fuel for natural selection. So permit a moment of speculation. Bacteria have circular genomes and plasmids, and every cell is a unique individual. Daughter cells don't have to have the same DNA as mother cells, and the species evolves quickly, which is why we deal with antibiotic resistance. But plants and animals, multicellular organisms, develop chromosomes so that every cell in the body has the same DNA. It's as if cancer pulled an ancient trick by making these circular particles driving oncogenes to drive variants. So we began to notice something. I began to notice that you're getting a ton of transcription from these, more than you'd expect just based upon the copy. You'd say, look, more gene, more template, more protein. But even more than you would expect when you normalized for copy number, we're seeing tons of transcription. At this time, I gave a talk at Stanford, and Howard Chang had just developed his incredible technologies to look at chromatin architecture. And Howard and I talked after the lecture and realized we're actually looking potentially at the same phenomenon. And that started a working relationship that continues to this day. It brought me to Stanford. We have a very unusual working relationship. And everything I'm going to tell you now, Howard is really the best experimentalist I know. And everything I'm going to tell you is work that we've done together going forward. But we realized we needed to understand the chromatin architecture of extrachromosomal DNA. And so we found a number of surprising things. First, let me just show you that, in fact, these really are circular pieces of DNA. There's no inference here. There's not just reconstructing reads. There, you see it. If you actually look at the chromatin accessibility on the top right using ATAC-C, which is a variant of ATAC-C, you can just visualize it. You can see that's a ton of accessibility on the extrachromosomal DNA particles. And in fact, when you did ATAC-C, we normally can see what's open, but you can actually learn something by looking at the pitch: monomers, dimers, trimers, tetramers, pentamers, and so on. And what we noticed was that, in fact, the DNA, extrachromosomal DNA, is chromatinized, but the very long patterns, the highly compact regions, are deficient in the extrachromosomal DNA. It's like having a can of tennis balls where the middle ball is missing and everything is open. It doesn't hypercompact. So it's highly accessible, and it's loaded with accessible chromatin marks and has a paucity of repressive chromatin marks. At this point, the Cancer Grand Challenges program that's sponsored by the CRUK and the National Cancer Institute nominated extrachromosomal DNA as one of the grand challenges. And I was fortunate to be able to bring together a team of people of very broad disciplines: Howard Chang, Vineet Bafna, Ben Cravat, Miriam Jamal Hanjani, Serena Nick Zeidel, James Chen, Andrea Ventura, and others. People from very diverse backgrounds to really tackle this problem. And what we really wanted to do was to maximize our integration, to have the trainees drive our ventures, to develop a set of new technologies or existing technologies that we could use in new ways to interrogate ecDNA, to learn from patients, and to go science to medicine. And I want to tell you about some of the things that we found. One of the first things we found, this is the work that was led by King Hung, a graduate student in Howard's lab. We were able to show that these ecDNA particles don't work alone. They interact with each other in trans. They form these hubs. An enhancer on one can talk to a promoter on the other, bridging the transcriptional machinery to become transcriptional factors. A second thing that we learned had to do with the problem of resistance. I kept wondering about this issue about why it was that these cells were more resistant. And then this hypothesis is this idea that you would get cell division that would be unequal in terms of the particles. So if you were actually to do a model of what would happen in the cell division if it's on chromosomes, each daughter would get half. But you would predict a bell-shaped curve, or essentially a random distribution of extrachromosomal DNA particles. So I actually tested that. We did an experiment in which we actually looked at two cells that are divided using Aurora B as the midbody and counted the ecDNA particles that went to each daughter cell. And independent of cancer type or oncogene, they all did the same thing. They followed the rules of random segregation. So then we also built a system in which we could image these in live cell imaging, and they did the same thing. It was random segregation. And this is why it matters. We took advantage of some isogenic pairs in which all, let's say, 100 copies were on extrachromosomal DNA, and then a subline in which they'd all jumped onto chromosomes. Same copy number, isogenic, but what was different was their response to treatments. If you withdrew glucose, for example, the chromosomal cells died, but the ones in extrachromosomal DNA didn't. Or if we treated with an EGF receptor inhibitor, the same result. How was that happening? And the answer was because it was changing its genes when it was on extrachromosomal DNA. If we looked at the copy number of the EGF receptor, the bell-shaped curve allowed selection at the bookends, either up or down, so that it could change its genome within 2 cell cycles. And we saw this in patients. This is really what was actually happening. So effectively, extrachromosomal DNA was really hitting 3 of the critical pillars of Darwinian evolution. Heredity, instead of identity by descent, it was becoming random identity by descent. The variation was markedly increased over chromosomal inheritance, and we had the ability to rapidly change genome because you could select on the edges. We then began to ask the question, I showed in that picture Darwin's branching tree of life. And when we think about how cancers evolve, typically people have used that as an inspiration. So you look at mutations typically, and you say, oh, you see it here, here, and here, and here, so it must have been truncal. Or if you see it here, but not there, but here, that must be a subclonal event. But Darwin was studying finches, he wasn't studying bacteria. That tree would have looked very different if it was non-chromosomal inheritance. So realized the best way to prove this point would be to actually look at patients who had a precancer that went to a cancer. And I'm very glad to have heard Dr. Schaefer's speech this morning about Barrett's, because this sets it up beautifully. We looked at those patients with Barrett's. We had 2 data sets, one from the Fred Hutch Cancer Institute, where they took half patients who went on to have cancer from Barrett's and half patients who didn't, and they were sampled over space and time. And we also looked at Rebecca Fitzgerald's cohort that was a large surveillance cohort of patients that either had Barrett's alone, Barrett's with this high-grade dysplasia, or cancer. And what we found, and it's, I think, I'm showing this was from the news and views of our paper. Tim Wang did this cartoon, and I think it's a wonderful cartoon because it illustrates the point. What we found was that extrachromosomal DNA could arise in Barrett's in high-grade dysplasia, always in the context of a p53 loss. And if it happened, those people would go on to get cancer, which raises the issue that we have to begin to think about ecDNA as potentially being involved in the development or the early stage of cancer. We also know it can happen later in cancer. So at this point, what we knew was that extrachromosomal DNA affects children and adults and women and men. It's not seen in normal tissues. Remember, this is not like the small circular DNAs that we normally have. These are large, a megabase or more. They're just absolutely huge. They're highly amplified, and they contain lots of genes and regulatory regions. They drive intratumoral heterogeneity because of this random segregation mechanism. They change how oncogenes are expressed, both by the regulation in cis and the regulation in trans, as those loops actually bring enhancers and promoters together, now turn it into a circle, and everything interacts with each other, and it drives rapid evolution to promote resistance. And we know it can occur pre-cancer or later in cancer. So it's a big problem. And then trying to understand that, I'm now going to tell you about 3 pieces of work that are coming together. They are all in the proof stages right now and will be out together soon, that I think show a kind of complementarity of what's happening in the field. The first was we needed to begin to understand how common is this? And the work I'm going to talk about now came from a close collaboration with Charlie Swanton and Miriam Jamal Hanjani, spearheaded by Chris Bailey and Oriel Pitch, using the Genomics England cohort. This is a fantastic cohort of close to 15,000 people with cancer. And we were able to actually use this Amplicon Architect tool from whole genome sequencing and ask the question, when does ecDNA arise and whom? This body map image shows you the frequency. So the Genomics England cohort is sort of biased a little bit towards the earlier stage 1 and stage 2 tumors, but it does have stage 3 and stage 4 tumors. And even so, 17.1% of all cancer samples had extrachromosomal DNA. And there were a few surprises in here. The frequency in breast cancer was far higher than we would have anticipated. But you can see here, many of the most aggressive forms of cancer are loaded with extrachromosomal DNA. There was also another very important surprise, which is that extrachromosomal DNA can come in multiple flavors. Not only do you have oncogene containing extrachromosomal DNAs, but you actually have immunomodulatory genes on extrachromosomal DNA, either those that are close to the oncogenes that are being on the extrachromosomal particles that are being co-selected, or circles themselves with immunomodulatory genes. And we also see circles that are regulatory elements only. And that makes sense in the context of these kind of cooperative interactions. When you actually looked, or when we looked at what kinds of immunomodulatory genes were being expressed, we even saw ligand receptor pairs that might make sense in downregulating the immune response. It's known that a lot of these patients with gene-amplified cancers don't respond to immune checkpoint therapy. And some anecdotal data on patients with extrachromosomal DNA increasingly suggests that that's the case. I think this begins to tell us that it's actually affecting the tumor microenvironment, the tumor immune microenvironment. And when we looked at inferred T-cell fractions, having these particles actually resulted in lower inferred T-cell fractions inside these tumors. We also, because this was such a large sample set, we could look at links to tumor suppressor losses. And gratifyingly, as we'd seen on our Barrett's paper, p53 loss seems to be a very common event that's tightly linked to the development of extrachromosomal DNA particles. We also were able to ask the question about metastasis. And I think what you can appreciate here is that it is tightly linked to the development of metastasis in specific cancer types. So this is a bad, bad problem all the way across the board. And most importantly, the clinical correlates of extrachromosomal DNA. It's very tightly connected with increase in stage. It's associated with chemotherapy resistance, targeted therapy resistance. It looks like there's something going on the immune therapy as well. And because the sample set was large enough, we were able to compare patients who had extrachromosomal DNA to patients who had other types of amplifications or no amplifications at all. It's clear that these patients do worse than all other cancer patients. So this is, again, a huge clinical problem. So then the next thing we began to work on is to ask, could we target this? I'm going to tell you about work now that is in the space of a synthetic lethality that we identified that we can hit with conventional chemistry. We're going to move on to non-conventional chemistry, working with Ben Cravatt and colleagues, but I want to tell you about conventional chemistry that's taught us something very important. So remember I mentioned about this highly accessible chromatin, and that you might predict, right, if you have highly accessible chromatin and active histones and lots of template, that you'd be getting a ton of this transcription while it's trying to replicate. So you should be getting transcription replication collisions, and that should actually be potentially very problematic to the tumor cells. So I'm going to tell you about work that was done again between my labs and Howard's lab, Jun Tang from my lab, Natasha Weiser and Weiping Wang from Howard's lab, amongst others. And what we did was we wanted to ask, what is the impact of this kind of rapid transcription? So we did a number of things. We did GRO-seq, RiboZero, and Cas-seq. Cas-seq is a way to look at single-stranded regions of DNA. And what you can appreciate, first of all, on the right, is there's a ton of this transcription, a ton of nascent RNA, and a ton of single-stranded DNA that's on these extrachromosomal DNA particles. And again, I should mention, this is using the COLA 320 system that's got MYC highly amplified in extrachromosomal DNA with an isogenic version in which it's jumped off onto chromosomes, HSRs, or homogeneously staining regions. If you look at the bottom and you see those tracks, you can also appreciate that there's these single-stranded DNA regions that you have on the extrachromosomal DNA particles. So all of this suggests that this ramped transcription is driving the accumulation of single-stranded DNA. So you would predict then that this would lead to a ton of replication stress. So first we just looked widely and we looked at the Cancer Genome Atlas samples and used two different signatures. I'm just showing you one of replication stress. And the tumors that have extrachromosomal DNA have far higher levels of replication stress. But I think maybe even more convincingly, Sudhir Chowdhury, who had been my former postdoc, is now at Boundless, did an experiment. We saw the fiber assay earlier today where you do the IDU-C-IDU to actually look at the loading of nucleotides onto growing replication forks, and you can assess the speed. But to that, we also added the FISH probe MYC so that we could actually look at it on the circles themselves. And what we noticed was that the loading of the DNA was slower, of the nucleotides was slower at the replication forks on the extrachromosomal DNA particles, very much consistent with this idea that they were under replication stress. So then we visualized it using phosphorylated RPA2 at the S33 site, and we located it on the extrachromosomal DNA particles themselves using this colocalization approach. And in each one of the isogenic paired cell lines that we used, you could see that you have a ton of replication stress happening on the extrachromosomal DNA particles themselves. Importantly, if this is driven by these collisions, blocking transcription should abrogate that. And we found that blocking transcription initiation with triptolide actually does that, and it does that specifically on the extrachromosomal DNA particles and on the EDU positive cells. So it really is the interaction between transcription and replication that's driving this enormous amount of stress. So you predict that there's be a lot of damage on these extrachromosomal DNA particles. Phil Hanoian and Steve Benkovic did this COMET FISH assay, and you can actually see here there's a ton of damage on these extrachromosomal DNA particles. And then we look at markers of DNA damage and also of the S phase checkpoint, check 1, all of these things are up on the ecDNA particles themselves. So this raised a very interesting issue. In terms of time, so Howard Chang and I and Ben Cravatt are all co-founders of Boundless. And Boundless Bio had identified check 1 in a genetic screen, but we didn't share that with our labs. We're very hygienic. But the people in our labs said, wait a minute, our data here says that if you need this S-phase checkpoint, if you abrogate it, they're going to go through into the cell cycle and get an enormous amount of damage, and they should be very sensitive to check 1 inhibitors. So do the experiment. And in fact, that was exactly what we saw. They'll go into that and you'll start getting DNA damage and cell death in the ecDNA containing cells, preferentially to the chromosomal. So then we realized that there was an opportunity to do something really interesting. So Boundless had developed oral CHEK1 inhibitors, and these are the data using a highly bioavailable oral CHEK1 inhibitor. And this is in a model in which we actually tested, it's got an FGF receptor amplification. And using infigratinib or the oral CHEK1 inhibitor or both, you can see the results, which is that you use the FGF receptor inhibitor, you get a partial response that fades pretty quickly. You use the CHEK1 alone, you get some response, but you put them together and you have a dramatic response. And the data on the right explains why, which is that's the copy number of the FGF receptor. And so effectively, the problem with targeted therapy when you've got extrachromosomal DNA is that the tumor can resist it by changing the copy number of the oncogene that you're targeting on extrachromosomal DNA. This is more like putting it in a vice grip, and it prevents that. And now they become sensitive to the targeted therapy. So at this point, I think we're adding to the list of what we know. We now know that ecDNA is very prevalent. It's linked to metastasis. It's linked to poor outcome and to therapeutic resistance. We know that there are actionable mechanisms, and this is likely just the first. And it can be amenable, we think, potentially to conventional chemistry, but we're also working very closely with Ben Cravatt to work on developing the kinds of new chemistry, because we imagine many ecDNA proteins of interest are going to fall in traditionally undruggable classes of proteins. And we're not done with surprises. We're starting to continue to, or we're beginning to continue to see how ecDNA breaks rules that we would expect. And I'm going to show you, I mentioned that there were 3 papers. This is the 3rd, and I'm just going to give you a teaser about it. So remember I had mentioned that ecDNA undergoes random segregation like a bell-shaped curve. And I also mentioned that 2 ecDNA particles can interact with each other in trans. One thing I also didn't mention is that about tumor cells can have more than one ecDNA flavor, including 2 ecDNAs with different oncogenes that could potentially interact. If you have an enhancer on one talking to a promoter on another and it's productive, you would imagine that the tumor would want to keep the winning hand. But how would that actually work in the context of random segregation? So we did the experiment, and here are just some data you can see at the top. This is a SNU16 gastric cancer cell line that's got both that's got FGF receptor and MYC on different ecDNAs. We did single cell cloning to make sure that these were not on the same circle. These were on different circles. And then asked the question, are they co-inherited? And the answer was, yeah, they actually were co-inherited. So then what is the mechanism? And I should mention, this is work, again, by King Hung and Matt Jones in Howard's lab and Ivy Wong in my lab. And this was probably the critical piece of data that turned it. How does this actually work? So this is looking at nascent MYC transcript in these cells. And that's in gray. And I think what you can appreciate here is that the ecDNA continues to transcribe as it goes through mitosis. That's not happening when it's on chromosomes or in cells that don't have ecDNA. That's not supposed to happen, but it continues to happen. And we actually identified transcription initiation using conventional chemistry, but also using Ben Cravatt's covalent chemistry to identify transcription initiation as a critical step. It is as if the interaction between these particles bridged by transcription is driving their ability to be co-inherited. I told you quite a number of things. And I want to point out that none of this would be possible without the way that we actually work together. When I realized the scale of the problem, or how important this actually was, and realized that if we're going to go from science to medicine, it would require a lot of expertise from really brilliant people, I reached out to an extraordinary set of colleagues, and we all rolled up our sleeves to share the problem and to try to solve it together. That's Howard Chang, Vineet Bhofna, Ben Cravat, Charlie Swanton, Miriam Jamal Anjani, James Chen, and others. And it's really taken a leap of faith from the people in my lab to work differently, because they work across labs now. They share credit, they share everything. And the Cancer Grand Challenges program that had the vision to understand that that kind of science requires being funded in a very different type of model. And to my home, Seraphin, Ken H., and the pathology department at Stanford that have really supported this kind of work, supported me. Thank you.
We have time for questions.
Really fascinating talk. I'm curious if you've looked at, since there's DNA obviously in extrachromosomal DNA, and these are continuously inherited, subject to the same pressures, do you see further mutagenesis in ecDNAs? Are you seeing signatures of selection, say APOBEC-like mutagenesis and selection for certain variants as cells are treated, continue to evolve, and metastasis maybe?
Yeah, absolutely. Actually, it's like a foundry for experiments, having that many copies. There are a few other things I didn't tell you. There was a paper from Lyudmil Alexandrov a couple of years ago. We worked with him on it. Tons of APOBEC-mediated mutagenesis going on in ecDNA particles. Part of this Genomics England paper that's about to come out, we also looked at mutational signatures, and there's a very strong APOBEC-mediated mutational signature. Interestingly, tobacco smoke seems to have a very strong impact on this as well. But we're quite sure, and actually, once they've formed HRD, HR deficiency seems to be involved. We think it is an ongoing mutagenesis problem. It's a very good question.
Thank you.
So we have time for one more question. Sorry.
Thank you for a very interesting talk. So I have a question, because recently I heard there's a talk about this chromosynthesis.
Yeah.
Chromatin can spontaneously break into hundreds of pieces. People found the tumor heads that actually they are less responsible to the immunotherapy. So this is kind of a paradox. Why they have so many pieces but not immunogenic? Does this actually relate to some ecDNA that can regulate the immune system?
Oh, I think it does. You've raised a very good point. So how does ecDNA form? Right, I didn't talk about that, but we do know something about that. So David Pellman's work, Peter Lee, Ofer Shoshani, Don Cleveland, they've had this model of chromothripsis, and that I think that does happen. And our data suggests that happens in about a third of the cases. Peter Park and David Pellman also have this work about transcriptional bridges. But there's also another route. We have data that says that paired double-strand breaks in the context of suppressor losses is a very potent way to do it. It is as if ecDNA is an end state here that's being linked to all of these potential routes.
Thank you.
You're welcome.
Right, let's thank Dr. Mischel once more. Thank you. That was fantastic. So our next speaker is Dr. Rameen Beroukhim, who's coming from the Dana-Farber Cancer Institute and Harvard Medical School. Dr. Beroukhim is a practicing neuro-oncologist whose research is really focused to understand tumor evolution with an emphasis in brain tumors and alterations to chromosome structures. So his work really spans computational methods development, genomic studies of human cancers, and also the development of novel model systems. And he has contributed tremendously to the development of several genomic analysis methods, and he has led the integrated genomic profiling efforts in multiple cancer types, including pan-cancer analysis across thousands of tumors. And this work has identified novel mechanisms by which cancers develop and become resistant to therapies. So please help me welcome Dr. Beroukhim. Thank you.
Thanks so much. So for that introduction and for inviting me to this fantastic conference. You know the feeling when you're sitting 10 minutes before you're about to give a talk and you realize, I'm giving the wrong talk. I should do a different talk. Well, I was going to give this talk about positive negative selection cancer genomes, and then I heard Paul speaking, and I thought, double extrachromosomal DNA, double minute MDM2, I should be giving this talk, P53 reactivation and GBM. So I threw that together in the last 10 minutes, and here we go. So one of the reasons I did that is because, Paul was talking about extrachromosomal DNA, but really he's a GBM guy, a glioma guy, right? So, gliomas, and I should say disclosures, I consult for folks and all that sort of thing. Gliomas really are the only important cancer, right? They occur in about 20,000 people a year, and they occur in the brain, which is an extremely horrible place for a cancer to occur, right? As soon as you're diagnosed, you know that your brain has been messed with. And they're basically universally fatal, right? And with glioblastomas, on average, killing folks within 15, 18 months. Despite our best treatments, which are surgery, radiation, and chemotherapy, the chemotherapy being standard alkylating agents, which haven't really changed for 20 or 30 years, right? So we desperately need to do better things for glioma, right? And the problem is, well, we've tried, right? We've done over 1,250 interventional glioblastoma trials since 2005. And I count that about 2 led to positive outcomes, right? And Paul may disagree with me there, but bevacizumab was approved and Tumor Treating Fields was approved. Tumor Treating Fields being very controversial. So the vast majority of these clinical trials failed. And we don't know why in the vast majority of cases. So what happens? You do a clinical trial of an agent, it fails, you don't know why, and then so you come up with another trial of a different agent or the same agent under different conditions or something, and you're flying blind, right? So, this is something that we've been thinking about a lot, about how do you not fly blind? So, in general, why do treatments fail, right? Well, one reason is pharmacokinetics. In the case of brain cancers, maybe the drug didn't reach the target, poor blood-brain barrier penetration, right? A second reason could be pharmacodynamics. The small molecule, say, didn't hit its molecular target. Maybe you hit target properly, but the cancer cells just didn't care and they didn't die. Or maybe you killed a bunch of cancer cells, but the tumor just regrew within a month or two, and secondary resistance arose quickly. Each of these, is figuring out each of these is important because any of them would require a different response, right? If you're just not getting into the brain, well, maybe you should find a way to get into the brain better. If you're hitting your molecular target, but the cells don't die, well, you have to rethink the therapy. And to understand those different consequences, you can't just look at the MRI scans, which is what we do. You really need tissue to distinguish between them. So most clinical trials don't get tissue after treatment, but that's what we need. So I'm going to take a step back and talk about a study we did a few years ago, which we submitted years ago and still haven't published. And so I guess this is a new data in terms of publication, where we took 78 different glioma models, both standard cell lines and patient-derived models grown in stem cell growth serum, or growth media without serum. And we did a whole slew of characterizations, looking at the genomes, the transcriptomes, the proteomes, and we treated each cell line, each of these 78, with over 400 different drugs at various concentrations to try to see which cell lines were responsive to which drugs, right? We integrated those data to figure out what genetic or genomic features predicted response, and then compared to sort of human data and other cancers. So just to cut to the chase, the best prediction of that we could find of treatment success or failure from the genomic data was of MDM2 inhibitors. And this is something that others have seen in other cancer types, that basically if you have p53 mutations, then the cell line will not respond to MDM2 inhibitors. If the cancer does not have p53 mutations, it tends to. And this is true for glioblastomas across the 78 gliomosphere lines. It's also true for other cancer types, right? So, that would argue then that maybe we should be using MDM2 inhibitors in clinical trials for p53 wild-type gliomas, right? So we've done that trial, or actually we're doing that trial. We've been doing a trial with ABTC and now the Alliance for Clinical Trials of p53 reactivation with the MDM2 inhibitor navtemadlin. And here we take patients and randomize them to 1 of 2 doses of navtemadlin. We treat them and then do a surgical resection, make sure that their tumor is p53 wild type, and then go on and treat with more navtemadlin. And the key point is that we do the surgical resection here, as you'll see later. So I want to talk about this trial. And I first want to talk about the people who actually did the work. So actually, if you think about it, who knows you best? It's your children, right? So my kids think I'm totally incompetent. And whenever you see me talk about anything, you know that somebody else did the work. So Veronica Rendo is a postdoc in my lab. Callum Quant Lee is a clinician scientist who ran the clinical trial. Keith Ligon is a pathologist and who I work with closely. And Patrick Wen is a fantastic clinical trialist as well. And so they did all the work that you can see. And this is the outcome of the trial, right? Overall survival was 15 months on the order of, well, actually 9 to 15 months or so on average. Progression-free survival, meaning the amount of time that the drug really appeared to work before the tumor came back or grew, was on the order of 3 months. Basically, we would get an MRI as we started the trial, and then treat with navtemadlin, and then 3 months later get another MRI to see how things were going. The tumor had grown, and we'd move on, right? So it's basically immediate progression. And I should point out that this is navtemadlin as a solo agent. There's another arm of the trial which is using it in combination, which I'm hoping will do better. But here we have a clinical trial where we're seeing things that don't really work very well. So again, going to the structure of the trial, there was this on-treatment biopsy that was being done, right? So this is something that is being done increasingly, fortunately, window of opportunity trials, where you give the drug for a little while and then you do a biopsy. And one of the reasons for the biopsy is to figure out, did the drug actually make it into the brain for brain tumors? But we realized that you can do a lot more things than just that. So again, we could ask all of these questions of that tissue that was obtained on the biopsy. Well, not all the questions, actually, not secondary resistance. But we could address 3 of the 4 questions with that initial biopsy after a couple of days on the drug. And then in some cases, we were able to get tissue from later on from autopsies, unfortunately, or other trials that were done later on these patients, where we could see what happened for secondary resistance. And so I'll walk you through what we looked at. So first of all, we did, Natalie Agar did imaging mass spectrometry of the tumor tissue, and essentially found that navtemadlin concentrations reached 137 nanomolar in contrast-enhancing tissue, basically the main tissue of a glioblastoma, and even the tissue on the edges of the glioblastoma where the blood-brain barriers looked really most intact, we got almost that high level of navtemadlin. The aim of the trial was to get over 50 nanomolars of navtemadlin concentrations, and so trial made the actual primary endpoint. So we found that that was not the problem. So the next question is pharmacodynamics. Well, the point of an MDM2 inhibitor is to reactivate p53. We saw that there were changes in MDM2 expression that indicated feedback mechanisms from reactivating p53. We saw increases in DDB2, DNA damage repair genes that also indicated an activation of p53, cell cycle arrest genes, and to some extent, apoptosis genes, BAX and PUMA, all significantly increased on navtemadlin. And this is relative to actually standard of care tumors, so treated tumors. So when you look at tumors that were resected after, these tumors had been previously treated with radiation and chemotherapy, as I'd indicated, and then this was a follow-up trial when the tumor had come back. So we compared these to tumors that had come back but had not been treated with navtemadlin, and there were these significant differences, right? So we can argue that pharmacodynamics was not the problem, right? So then, well, is there a cellular response? Well, actually, I'll just jump to the fourth thing. The mechanism of resistance to MDM2 inhibitors in most cancers is thought to be p53 mutation. If you inactivate p53 directly, an MDM2 inhibitor won't work, right? Because MDM2 inhibitors work by increasing p53 protein levels, and if the protein is inactive, who cares? So we actually did have 3 patients who had post-progression biopsies. Again, I think at least 2 of these were autopsy cases, unfortunately. Not a single one of them showed mutations in p53. So all these tumors had gotten a pharmacodynamic response, but recurred anyway, or grew through the treatment anyway, without having p53 mutations, which is the most expected secondary resistance mechanism. So, right, so then the last thing to look at is the cellular response. And what we found was that, in fact, the cellular response was insufficient. That when we treated with, when we used similar doses of navtemadlin in vitro to what we used to what was seen by the tumor in humans, we got on the in sort of glioma cell lines that are p53 wild type, we got on the order of 30% apoptosis, right? So killing some of the cells, but most of the cells were not dying. You can imagine 30% apoptosis in a cancer in the brain is not going to really do that much. We tried using increased doses of navtemadlin for shorter periods. That actually did worse. We looked at bone marrow cells because navtemadlin's main toxicity is bone marrow toxicities, and actually we saw very little happen there, which was nice at these concentrations, but we were ineffective. Why were we ineffective? Well, one possibility is cell state changes, and we heard a great talk from Alex Shalik earlier, and who knows much more about cell state changes than I do. But we did sequencing on the tissue before and after treatment, both on the patient biopsies and on our cells that we treated in vitro. And in the biopsies, for example, we found a whole slew of genes that were overexpressed post-treatment, on treatment compared to pre-treatment. And those tended to point to cell state changes with a slightly more differentiated cell state, in fact. So basically going from sort of a progenitor state to a more transit amplifying type of cell state. And that was different from control samples again. So again, here, this is single cell data from cell line treated with navtemadlin, where we see, again, increases in cluster 3, which represent sort of more mature oligodendrocytes relative to progenitor-like cells, like in cluster 1 or so on. So that may have contributed. So then the question is, well, how can we get better responses, right? Could you do higher doses of navtemadlin? Would that do it? Or could you do a combination therapy? Again, we would have no idea if we didn't know what was going on in the patients. Now we had an idea that we were getting, maybe some pharmacodynamic effect with a halting of cell cycling to some extent, but no apoptosis. So if you go to higher doses, how high of a dose do you have to get to really full apoptosis? Well, for monotherapy, with one of our p53 wild-type lines, we had to go to about 500 nanomolar naftamadlin and continuous dosing to get to really complete apoptosis, right? And so that's really high. That's something that we would never be able to attain in humans. So the next question is, well, how about we use combination therapies, right? P53 reactivation works by taking advantage of replication stress, right? The cells are dividing, their DNA is screwed up, and P53 notices that their DNA is screwed up and that they're trying to divide too quickly, and it kills them. So can you increase replication stress and cause more cell death? Well, the standard treatment for glioblastoma is temozolomide, standard chemotherapy. Well, actually, there are two treatments, radiation and temozolomide, and both of them increase replication stress. Arm 2 of the trial is a combination of naftamadlin with radiation. So we decided to test temozolomide as well. And in fact, what we saw is that for both of these lines, the combination of naftamadlin with temozolomide led to decreased growth relative to either agent alone. We also found that these two agents had non-overlapping resistance mechanisms, meaning that cells that had gained resistance to temozolomide were not resistant to naftamadlin. And so the main resistance mechanism to temozolomide is loss of mismatch repair. We reproduced that with isogenics that had lost MLH1 or MSH2, and those had just as much sensitivity to naftamadlin as the GFP controls. And I'm pointing at the laser at this screen that none of you can see. Whereas P53 loss, led to full resistance as we would expect it. When combining temozolomide and naftamadlin, we saw additive effects in glioma spheres, right? The P53 wild-type glioma models, with these two-dimensional charts, as you can see, with increasing doses of temozolomide on the horizontal axis and increasing doses of naftamadlin on the vertical axis, where we could get to pretty high rates of apoptosis at sort of combinations of naftamadlin around 100 nanomolar with temozolomide doses up at around 10 micromolar range, which are actually not that high for temozolomide. Now again, both of these treatments actually have effects on the bone marrow with thrombocytopenia in particular, loss of platelets, right? Whenever you do a combination, whenever you combine treatments to try to get a better effect, of course it's going to have a better effect on cancer cells, but the question is about therapeutic window. And so we tried this combination on bone marrow stem cells, and we saw essentially very little additive effects, really no apoptosis due to the combination. So that's a sort of good sign that this sort of thing could be potentially feasible in humans. So that's the summary of what I would talk about, which is that MDM2 inhibitors are likely most effective in combination with other agents, but more to the point that really the clinical trial paradigm should be moving much more towards getting on treatment and post-treatment tissue sampling if we're going to want to make any sort of informed decisions about how we're going to do the next trials and how to improve treatment. And to that end, I want to point to Breakthrough Cancer, where actually Michael Simo was here in the audience earlier, but told me that for my talk, he was going to get the hell out. And the reason he was going to get out was because he's actually co-leading the glioblastoma effort for Breakthrough Cancer, where we have dozens of laboratories and clinicians focused on developing a new clinical trial paradigm, which involves longitudinal sampling, multiple surgeries to extract brain tissue, tumor tissue, while people are receiving treatment to see what's going on with those treatments. And so hopefully we'll have more information for you from our first trial soon. And with that, I'd like to thank the folks in my lab who contributed to this, including Veronica Rendo, Ruben Ferrer-Luna, and others. Keith Ligon, who's my partner in crime for a lot of things, and my fellow clinicians at the Dana-Farber, including Callum Lee and Patrick Wen, who sort of ran this trial. And as was pointed out earlier by, I think, Alex, our patients and their families who, are giving their lives for this. And I want to thank you, and I'm open to questions.
Questions? We have plenty of time.
Great talk. I didn't quite get the timing, but given the feedback mechanisms and how long it may act, how about combination? How about intermittent therapies? We look at the kinetics of what happens, induction of death, induction of p53, and then, given that kinetics, create a...
Right.
So, just as an editorial, no one...
Yeah. So it's a great question. So what you're sort of... so the way the treatment is given is like 1 week of navtemadlin and then 1 week off, because if you gave it continuously for 2 weeks, bone marrow would, not make it. So I think what you're suggesting is why not pulse it with very high doses and intermittently at sort of lower frequency. Is that?
What was the effect on people last if one dose?
Yeah. So I think so the question is, well, how long does the effect of p53 last when you give one dose? I think it lasts as long as the drug is there. Essentially, so MDM2 degrades p53, and so as soon as the drug is off, MDM2 goes back to degrading p53. The drug is dosed daily, which means that its half-life is on the order of a day. So you could try pulsing at very high doses, but when we tried that, we actually got worse outcomes in terms of apoptosis. So I think more continuous dosing, might work, but we'd have to be at levels,
I guess what I'm asking is, does the induction of apoptosis tachyphylaxis continuous there?
That's right.
Tachyphylaxis, there's an effect on apoptosis die with time.
That's a great question. So again, does the effect of apoptosis die over time? It's not a clear. Sorry. Yeah, the question was, does the effect on apoptosis die over time, decrease over time? And we did not specifically test that. The amount of apoptosis that when we observed insufficient apoptosis, it was at around 5 days. I don't think it didn't. When we continue to grow the cells in navtemadlin, it's not like they got more sensitive over time. And anyway, you have to stop giving it after a while. So, yeah. Any other?
We have a question over there.
Great talk. So, do you think replacement of wild type p53 mRNA can alleviate treatment resistance?
Sorry, can you say that again?
Can we replace p53 mRNA?
Oh, can you use an mRNA treatment to replace p53? That's a great question. I think delivering nucleic acids, for instance, or any kind of large molecule to cancer cells has been very difficult. You need to get not 99% of the cancer cell delivery to not 99% of the cancer cells, but to fully 100% of the cancer cells, and that's been nearly impossible. There are folks like Joel Strela, who might be in the room, who have been working on a lot of folks here at the Koch Institute and MIT in general, who've been working on those kinds of delivery methods. I'm hoping that they can work, but especially when you have to cross the blood-brain barrier, it's been very difficult.
Thank you.
Yeah. Francisco.
Yeah, great talk. I'm going to ask two obvious questions after Paul's talk. First one, MDM2 being textbook example of extrachromosomal DNA. Did you see or did you look as to whether the resistant samples had just higher levels of MDM2 extrachromosomally? And the second one is, given that temozolomide is exploiting replication stress, do you think that there's a correlation in terms of response of having ecDNA, high replication stress, and response to p53 reactivation?
So I was thinking about your first question, and I didn't hear the second one. Well, let me answer your first question.
Replication stress.
Yeah.
That was the second question.
What about replication stress?
that since Paul just showed that ecDNA.
Oh, right. So in model systems, like kind of anecdotally, we see that MDM2 amplifications predict better response to MDM2 inhibitors. That might be what you'd expect. Because this is extrachromosomal DNA, it's hard to keep it sort of stable in the model systems, as Paul described. So yeah, we see that. And we also, in a couple of cases, we had patients with MDM2 amplifications who got this, and their amplification levels did go up in post-treatment biopsies relative to pre-treatment. But that's like 2 patients, and we know that this is extrachromosomal DNA is very heterogeneous across tissue. And so, what do I make of that? I don't know. And in terms of like whether the extrachromosomal DNA is leading to replication stress, that is, I imagine that's could be the case. So that, but there are many contributors to replication stress in these tumors, obviously.
Right. I'll ask the last question.
Yeah.
So I wonder if you have any insights on that oligodendrocyte differentiation phenotype that you saw transcriptionally, whether that could be a mechanism to promote this terminal differentiation as a therapeutic avenue.
Right. So that's a great question. So yes, I think trying to promote terminal differentiation would be really interesting. An interesting point is that Jeremy Rich has proposed that regular chemotherapy and radiation works best on slightly more differentiated cells, with the more progenitor-like cells being most sensitive. But then here we're seeing that for the MDM2 inhibitor, and actually in another study, BRD4 inhibitors, although questions about how useful those are, they both tended to migrate the cells more towards transit amplifying cells. So one question is whether again, like using temozolomide, since it's more active maybe in the transit amplifying cell population relative to the stem, more stem-like cells, is the opposite. Combining that with MDM2 inhibition. So that was one of the thoughts that was guiding us in that combination.
Excellent. Let's thank Dr. Beroukhim and Dr. Mischel once more.